Today we will talk about the DNA structure. DNA structure. DNA is deoxyribose nucleic acid. DNA structure, that is, DNA is deoxyribose nucleic acid. This DNA is a first important point regarding DNA structure is that DNA is a polymer of deoxyribonucleotides. So DNA, it is a polymer of deoxyribonucleotides. Polymer of deoxyribonucleotides. Nucleotides. This DNA is a polymer of deoxyribonucleotides. It means that these deoxyribonucleotides, they are the building blocks of the DNA. It is polymer because it is made up of thousands and millions of units, deoxyribonucleotide units. 4 types of deoxyribonucleotides are the building blocks of the DNA, or the basic units of the DNA. One is, we have deoxyadenosine, deoxyadenosine, deoxyguanosine, deoxycytidine. These are the building blocks, basic units of the DNA. Deoxythymidine, and these are the fourth one, deoxycytidine. These are the building blocks, basic units of the DNA. Or we can also write down that deoxyadenosine monophosphate is the basic building or the basic unit of the DNA, building block of the DNA. That is deoxyadenosine monophosphate. This is guanosine monophosphate. This one is deoxythymidine monophosphate, because monophosphates are the actual units of the DNA. Deoxycytidine monophosphate. So these 4 deoxyribonucleotides, deoxyadenosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate, and deoxyguanosine monophosphate, these are the building blocks of the DNA. DNA molecules are made up of these basic units. That is why it is called as polymer. And in a cellular DNA or RNA, we find hundreds, thousands, and millions of these deoxyribonucleotides in case of DNA, and ribonucleotides in case of RNA. So here we are discussing about the, if we'll see this deoxyribonucleotide, this deoxyribonucleotide. This deoxyribonucleotide, it is basically a nucleotide. It is basically a nucleotide. Now, in a nucleotide, we have got how many components? In a nucleotide, it has got 3 components. One is called as sugar component, another is called as phosphate component, and third one is called as nitrogenous base. So we can make it like this. For example, if this is what this is, if this is sugar component, sugar, then it is nitrogenous base, and this is phosphate. This is nitrogenous base. This is a typical, this is sugar, this is phosphate and nitrogenous base. This is a nucleotide. So we can say that we have 3 components. One is sugar component, sugar moiety, what is called as. Another is phosphate component. phosphate group and nitrogenous base. So in case of nucleotide, we have, this is a typical nucleotide. This is sugar component in it. This is phosphate group. This is nitrogenous base. So sugar moiety, phosphate group, and nitrogenous base. We will discuss now one by one. So we have, first we have sugar. We have sugar. This is pentose sugar. Pentose sugar. This sugar, in case of DNA RNA, it's of two types. One is ribose sugar, and another is deoxyribose sugar. Deoxyribose sugar. This pentose, it is 5 carbon sugar. 5 carbon sugar. That is why it is called as pentose sugar. The sugar present in DNA RNA is a 5 carbon sugar. It is called as pentose sugar. And in case of RNA, we have got ribose sugar, and in case of DNA, we have got deoxyribose sugar. So this is C5H10O5 in case of RNA, and this is C5H10O4 in case of DNA. That means, why it is called as deoxy? That means one oxygen has been removed in case of DNA, so it becomes deoxy. Deoxy means removal of oxygen. Actually, it is just a ribose structure, same as that of ribose sugar, but one oxygen present at the carbon second has been removed in case of DNA, so it becomes deoxyribose sugar. This deoxyribose sugar is found in DNA. So we will first take ribose sugar. This is so we have got 5 carbon atoms. So this is carbon is present here. Carbon is present. We can calculate. So 1, 2, 3, 4, 5 carbon atoms. Now we will just make the valency of carbon tetravalent. Carbon is always tetravalent. So we have OH, H. Carbon is having tetravalent. Carbon is tetravalent. It is having 4 valency in all cases. It is tetravalent. So 1, 2, 3, 4. This is complete. This is H, OH, H, OH. This is H, OH. This is the ribose sugar. Ribose, we can calculate how many carbons are 1, 2, 3, 4, 5. 5 carbon. How many hydrogen? Hydrogen will be 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We have 10 hydrogen. And how many oxygen? Oxygen will be 5. Calculate 1, 2, 3, 4, 5. This is ribose sugar. And this ribose sugar is found in RNA. Now we'll make a deoxyribose sugar. In case of deoxyribose sugar, the structure is by and large same, but one oxygen at the carbon second of the sugar had been removed. So we can say that this is pentose sugar. We have carbon 1, second carbon, third carbon, fourth carbon, and this is fifth carbon. 1, 2, 3, 4, 5. Same structure. OH, we have hydrogen. Carbon is tetravalent. We have to satisfy the valency of the carbon. In every respect, it should be tetravalent. So we have H. This is H. So H, this is OH hydroxyl group at carbon third. This is hydrogen here. We have CH2OH. So you can calculate, this is deoxyribose sugar, deoxy. Actually it's 2-deoxyribose. Why 2? Because oxygen has been removed at the carbon second. So deoxyribose sugar. So it is C5 1 2 3 4 5 carbon atoms. Then how many hydrogen atoms are there? 1 2 3 4 5 6 7 8 9 10 hydrogen atoms. How many oxygen atoms are there? We have 1 2 3 and 4. So this is called as deoxyribose sugar. Actually it is 2-deoxyribose sugar because of the fact that the carbon present at the carbon present at the oxygen which is present at the carbon second, it has been removed. So it is called 2-deoxy. That means oxygen present at the carbon second has been removed in case of deoxyribose sugar. So it becomes deoxyribose C5H10O4. So this indicates that oxygen removed at carbon second of sugar. Oxygen removed at carbon second of sugar in case of DNA, deoxyribose sugar. And this deoxyribose 2-deoxyribose sugar, it is found in DNA. So this was ribose sugar, another deoxyribose sugar. See, this carbon first, this carbon first, it is the site where nitrogen base gets attached to the sugar. So it is a site where this carbon first, that means at the carbon first nitrogen base is attached. So carbon first nitrogenous base is attached, and nitrogenous base is attached to the carbon first of the sugar by means of a bond that is called as glycosidic bond. So we have here the structure will be like this, glycosidic bond. So it would be like this. For example, we have here, this is glycosidic bond, this is nitrogen base, this is sugar, glycosidic bond. Actually it is called as N-glycosidic bond. N-glycosidic bond. Why N? It is basically nitrogen is a component of nitrogenous base, so we call it N-glycosidic bond. So here we have got phosphate. This phosphate, see, carbon first is the site where nitrogen base gets attached. So nitrogenous base gets attached to the carbon first. We can here write carbon first by means of what bond? Glycosidic bond. So here glycosidic bond is present. This glycosidic bond by virtue of the sugar is attached to the nitrogenous base. Then we have another type at carbon. These are some important sites, important positions. Carbon first of the sugar is important. Carbon third of the sugar is important because at the carbon third we have got hydroxyl group. We have got hydroxyl group. Remember. And at the carbon fifth we have got phosphate group. Carbon 5. This is carbon 5 of the sugar. Phosphate group is attached. Phosphate group attached. So we can here, this is carbon 5, this is carbon 5th, this is carbon 5th, and to this carbon 5th phosphate group is attached. So remember that in a DNA molecule we have got carbon 5, to this carbon 5 phosphate group is attached. In the DNA we have got 3' position, that means hydroxyl group is attached. At carbon first we have nitrogenous base attached. Nitrogenous base attached to the carbon first of the sugar by means of bond that is called as glycosidic bond, and the phosphate group is attached to the nitrogenous, the phosphate group is attached to the sugar component. The phosphate group is attached to the sugar component by means of a bond that is called as phosphoester bond. So here what type of bond would be there? Phosphoester bond. So this is for example carbon 5. This is carbon 5. This phosphate group is attached to the carbon 5 of the sugar by means of bond that is called as phosphoester bond. Phosphoester bond. That means phosphate is attached to the carbon 5th of the sugar by means of a bond that is called as phosphoester bond. So we have glycosidic bond by virtue of which it is attached, nitrogenous base attached to the sugar component. We have phosphoester bond. By virtue of this bond, phosphoester bond, phosphate group is attached to what? Sugar component. So then this was regarding the ribose sugar, deoxyribose sugar. Then we have another one, that is phosphate group. We have phosphate component, phosphate. It is derived from H3PO4, phosphoric acid. In cells, this phosphate group is derived from phosphoric acid, H3PO4. So we have this structure, P, OH, 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 double bond O. So this is the phosphate group which is found in this phosphate group is found in what? In this DNA or the deoxyribonucleic or the ribose nucleic acid. This phosphate group is present in DNA. Then we have got most important, that is nitrogenous bases. Nitrogenous bases. So we have nitrogenous bases. I will rub this. We have nitrogenous bases. Nitrogenous bases. Two nitrogenous bases are present. One is called as purine, another is called as pyrimidine. Two types of nitrogenous bases are present in DNA. One is called as purine, another is called as pyrimidine. So in a DNA molecule, two types of nitrogen bases are present. One is called as purine, another is called as pyrimidine. So this purine, what is the difference between the purine and pyrimidine? Remember that this purine, it is a 9-member ring. 9-member ring. It is a 9-member ring. So it would be like this. It is a 9-member ring. It is a 9-member ring. It has 5 carbon and 4 nitrogen. So it is a 9-member ring. This is a typical purine. This is a typical purine. Purine is a 9-member ring. So we have nitrogen here, 4 nitrogen atoms, in which we have got 4 nitrogen atoms, and we have got 5 carbon atoms. 5 carbon atoms. So it would be like this: nitrogen 1, 2, 3 and 4. So 1, 2, 3, 4, 5, 6, 7, 8, 9. So it is a 9-member ring where we have carbon atoms and nitrogen atoms. We will just fulfill the valency of the carbon. it is a 9-member ring. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 9-member ring where we have 4 nitrogen atoms. We have 4 nitrogen, 1, 2, 3, 4 nitrogen atoms, 5 carbon atoms. We have 5 carbon atoms. It has got how many rings? It is a double ring structure. It has got double ring structure. This is single, not a single ring structure, but it's a double ring structure. It has got double. Purine is all double ring molecules. We can say double ring structure. These are double purines are double ring molecules, and these purines are comparatively larger as compared to that of pyrimidines. They are larger. And we have 2 types of purines, that is adenine and guanine. Adenine and guanine purines. Then we have pyrimidines. Pyrimidines are 6-member ring. They are 6-member ring. They have carbon and nitrogen. It would be like this. It is how it is 1, 2, 3, 1, 2, 3, 4, 5, 6. So it is how this pyrimidine is a 6-member ring. The 6-member ring. It is a 6-member ring where we have how many nitrogens? Nitrogen atoms, 2 nitrogen atoms, and 4 carbon atoms. It is a single. It is a single ring molecule. So pyrimidine is a single ring molecule. Single ring molecule. It is a single ring molecule, and it is comparatively, this pyrimidine is comparatively smaller as compared to that of the purines. This is smaller because it is a single ring structure, whereas purines are double ring structure. Small. And we have how many pyrimidines are there in DNA? We have thymine and cytosine, whereas in RNA we have uracil. In RNA we have uracil. In RNA thymine is absent, but instead of thymine uracil is present in RNA. So this is the purines and pyrimidines which are found in nitrogens, which are found in DNA. These are the nitrogenous bases. Now we'll see how these nucleotides are formed. So in a DNA molecule is a DNA. It has 2 polynucleotides, 2 polynucleotide chains, 2 polynucleotide chains. This DNA is made up of 2 polynucleotide chains. I'll explain like this. This is one polynucleotide chain. This will be another polynucleotide chain. So these are 2 polynucleotide chains. These are 2 polynucleotide chains. It would be like this. These 2 polynucleotide chains, they are joined with each other. 2 polynucleotide chains are joined to each other by means of a bond that is called as hydrogen bond. So we have 2 polynucleotide chains are joined to each other by means of a bond. This is a bond by means of hydrogen bond. Hydrogen bond between 2 DNA strands. 2 DNA strands. And this hydrogen bond, which is the only attractive force between the 2 DNA strands, it provides a sort of stability to the DNA molecule. This hydrogen bond confers the stability to the DNA molecule. So we have got I will explain like this, these 2 polynucleotide strands, and remember that these 2 strands run in opposite direction. For example, we have 5 dash here, another 3 dash, this is 3 dash and 5 dash. That I will explain a bit later what this 3 dash 5 dash means. So the hydrogen bond exists between 2 DNA strands. So we have this structure. For example, this is phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. Obviously this sugar is attached to what? It is attached to nitrogenous base. For example, adenine. Sugar is attached to nitrogenous base. For example, guanine. Sugar is attached to nitrogenous base. And for example, cytosine. This is one polynucleotide strand. This is one nucleotide. This is another nucleotide. Fourth nucleotide. Fifth. This is one polynucleotide strand. Then another polynucleotide strand would be like this. This nitrogen base adenine always pairs with thymine. Thymine of the other polynucleotide strand. Guanine always pairs with cytosine. Adenine pairs with thymine, and cytosine pairs with guanine. So this is one strand. This would be another strand. This is nitrogen base is always attached to the sugar. So we have phosphate here, phosphate, sugar, phosphate, sugar, phosphate. So this is one strand, this is another strand, and this is called as hydrogen bond. There are 2 hydrogen bonds between adenine thymine and 3 hydrogen bonds between guanine and cytosine. 2 hydrogen bonds between adenine thymine and 3 hydrogen bonds between guanine and cytosine. So we can say that adenine and thymine, there are 2 hydrogen bonds, and guanine and cytosine has 3 hydrogen bonds in a DNA molecule. So this means this is one strand, this is one strand of the DNA, one polynucleotide strand, this is another polynucleotide strand, and these 2 strands are joined by hydrogen bond, and this hydrogen bond exists between nitrogenous bases. In nitrogenous base, that means adenine of one strand is joined to thymine of another strand, guanine of one strand is joined to cytosine of another strand, and adenine of one strand is joined to thymine of another strand by means of hydrogen bond. And this hydrogen bond gives stability to the DNA molecule. So this is hydrogen bond. Then we have another type of bond in the this is called a sugar phosphate backbone. In a DNA molecule, we have sugar phosphate backbone. This is called a sugar phosphate backbone, and this sugar phosphate backbone it has a bond that is called as phosphodiester bond. So this is called as sugar phosphate backbone, sugar phosphate backbone. And there is a bond that is called as phosphodiester bond. Phosphodiester bond. There is a phosphodiester bond. So one is hydrogen bond, another is phosphodiester bond in the sugar phosphate backbone, and the third bond which is present in a DNA molecule, that is glycosidic bond. And glycosidic bond that is present between the sugar and nitrogen base. It would be here. This is what is called as glycosidic bond. That is, this nitrogen base attached to sugar by means of a bond that is called glycosidic bond or N-glycosidic bond. This sugar is attached to nitrogen base by means of a bond that is called as glycosidic bond. So this is a glycosidic bond. So in a DNA molecule, 3 bonds are present. One is called as glycosidic bond. This is between sugar and nitrogenous base. Phosphodiester bond, it is present in the backbone, sugar phosphate backbone of the DNA molecule. And then we have another bond that is called as hydrogen bond, which exists between two strands. For example, if this is one strand, this is one strand of the DNA, this is one strand of the DNA, this is another strand. These two strands are joined by means of hydrogen bond, and which exists between the nitrogenous bases. And these two strands, this is, then we have the two strands are antiparallel. What it means? Two strands are two DNA strands are antiparallel. Antiparallel. What it means? It means that we have two strands. This is one strand, this is another strand. This would be 5 dash, this would be 3 dash, this is 3 dash, 5 dash. What this means? This is, for example, it's a double stranded DNA molecule. It's a DNA molecule. It has two strands. This is one strand, this is another strand. They are antiparallel. That means one strand moves in, one strand runs in 5 dash to 3 dash direction, another strand runs in 3 dash to 5 dash direction. It means that what this 5 dash means and 3 dash means, it means that 5 dash and means that the carbon present at the 5th position, the carbon present at the 5th position of the sugar has phosphate group, and that is free. That carbon, this 5 means carbon at the 5th position of the sugar, carbon at the 5th position of the sugar. So this, what do you mean by 5 dash end? The carbon present at 5th position of sugar has phosphate free. We know that. Remember that at 5 dash carbon we have phosphate, at carbon 5 we have phosphate, and carbon 3 we have what? We have hydroxyl group. This is very important to remember, that at carbon 5th of the sugar we have phosphate group, and at the carbon 3rd of the sugar we have hydroxyl group. So carbon at the 5th position of the sugar has phosphate group, that is free. That means in this case this phosphate group in this strand would be free, whereas in this strand the hydroxyl group present at the carbon 3rd of a sugar would be free. So this 3 dash end means that carbon at 3rd position, this carbon 5th means carbon 5th means 5th position of the carbon 3rd means 3rd position of the carbon in a sugar, which is third. At the 3rd position of the carbon of the sugar we have hydroxyl group. At the 5th position of the sugar we have what? We have phosphate group. So 3 dash end means carbon at 3rd position of sugar has hydroxyl group free. So it means that phosphate, if these are two strands, of the DNA molecule, phosphate present at this strand would be free, and hydroxyl group present at the carbon 3rd of this strand would be free. Then in the opposite, this is 5 dash, then it is opposite. That means hydroxyl group present at the carbon 3rd would be free, and in the opposite strand the carbon present at the 5th position would have phosphate group free. This is called as, so we call it that the two strands have opposite polarity. Two strands have opposite polarity. that means 5 dash to 3 dash, 3 dash to 5 dash. This is called as antiparallel. That means one strand runs in the 5 dash to 3 dash direction, another strand runs in the 3 dash to 5 dash direction. What do you mean by two strands of the DNA are antiparallel? That means one strand, if these are two strands, one strand runs in the 5 dash to 3 dash direction, one strand runs in the 5 dash to 3 dash direction, another strand runs into 3 dash to 5 dash direction. This is 5 dash to 3 dash direction and 3 dash to 5 dash. Why it is so? Why the two strands are antiparallel? Because replication always takes place in the 5 dash to 3 dash direction. To maintain the precise nature of this replication, the two strands of the DNA are antiparallel. That means they run in opposite directions. That is, one runs in 5 dash to 3 dash direction, another strand runs in 3 dash to 5 dash direction, so that it would help in the replication. Replication always takes place in 5 dash to 3 dash direction. So remember that replication always takes place in 5 dash to 3 dash direction. This is replication always takes place in 5 dash to 3 dash direction. That is why two strands are antiparallel. This is what do you mean by antiparallel. Then two strands of DNA are complementary. What it means? Two strands of DNA are complementary. Two strands of DNA are complementary. Two strands of DNA are complementary. It means that if we have got one strand of DNA and the base sequence in one strand of the DNA is, for example, CAT, AAT, CCA, GGA. This is the base sequence of bases in one strand of DNA. Then the sequence of bases in the other strand of DNA can be easily predicted because of the Watson and Crick base pairing rule. Cytosine always pairs with guanine, adenine pairs with thymine, thymine pairs with adenine, adenine pairs with thymine, thymine pairs with adenine, cytosine pairs with guanine, cytosine pairs with guanine, adenine pairs with thymine, guanine pairs with cytosine, guanine pairs with cytosine, adenine pairs with thymine. So we can say that if the base sequence in one strand of the DNA is known, we can easily predict the base sequence in the another strand of the DNA. So two strands of the DNA are complementary. That means if the base sequence in one strand of the DNA is known, we can easily predict the base sequence in the another strand. This means that the two strands are complementary. As we know that adenine always pairs with thymine or vice versa, and guanine always pairs with cytosine and vice versa. Adenine always pairs with thymine, thymine always pairs with adenine, guanine always pairs with cytosine, cytosine always pairs with guanine. So this is what is called as complementary. Then we have got, these are complementary. Now we'll see that this formation of nucleoside. What do you mean by nucleoside? Formation of nucleoside. How nucleoside is formed? Nucleoside is formed when we have got a sugar component. When nitrogenous base gets attached to the sugar component, we get a what? Nucleoside. So this is sugar component, and when nitrogenous base gets attached to it, how nucleoside is formed? By linkage of nitrogenous base to sugar by means of N-glycosidic linkage. By N-glycosidic linkage. Actually, it is the in case of purines, it is the nitrogen which is present at the 9th position. It is having linkage. It is having bonding with the carbon first of the sugar. Same is the case. The nitrogen present at the first position of the pyrimidine, it is having bonding with the carbon first of the sugar. So we sometimes use the term N-glycosidic linkage. So this is a nucleoside. This nucleoside is composed of two components. One is sugar and another is nitrogenous base. Nitrogenous base can be adenine, thymine, guanine, cytosine in case of DNA. Then how nucleotide is formed? Formation of nucleotide. In case of nucleoside, we have sugar component and nitrogenous base. And when phosphate group gets attached to the nucleoside, we get a nucleotide. So we have nucleotide. This is simply nucleoside. This is sugar. This is nitrogenous base. And when phosphate group gets attached, this phosphate group attached to the carbon fifth of the sugar, that we get nucleotide. This is nucleotide. What is difference between nucleotide and nucleoside? That means in nucleoside we have two components, sugar and what? Nitrogenous base. And in nucleotide we have three components, sugar component, we have nitrogenous base, we have phosphate group. So it is called as nucleotide. So how two dinucleotide is formed? Formation of dinucleotide. Formation of dinucleotide. Formation of dinucleotide. That means two nucleotides are joined by means of a bond that is called as phosphodiester bond. So we have got like this structure. This is one nitrogenous base, phosphate group. Then obviously this is at the carbon third we have hydroxyl group. It will be having bonding with the phosphate of another nucleotide. Then phosphate. This is what is called as, this is one nucleotide, this is another nucleotide, so it is a dinucleotide. And the bond here is phosphodiester bond. So what type of bond is here? Phosphodiester bond. We have phosphodiester bond between two adjacent nucleotides. So this is a dinucleotide is formed when two nucleotides get linked by means of a bond that is called as phosphodiester bond. We said that formation of dinucleotide. Same is the case. Hundreds, thousands and millions of nucleotides get joined and we get what? Polynucleotide. So we say that DNA is a polynucleotide. That means it is composed of hundreds, thousands or millions of nucleotides. So DNA has two polynucleotide chains. If we make one polynucleotide chain, it would be like this. That will also define it as polynucleotide. So we will make a polynucleotide here. This is sugar. This is sugar component, nitrogenous base. This is phosphate component. This is one nucleotide. Obviously at the 3' at the carbon third we have hydroxyl group. It will form bonding with the phosphate of the another nucleotide. Then it would be dinucleotide. This is dinucleotide. Obviously this hydroxyl group, it will form bonding with there would be a phosphoester bond with the adjacent nucleotide. So we'll get this structure. These are three nucleotides. What is called as in this way we can make the polynucleotide. So we have hydroxyl group attached to the phosphate of the adjacent nucleotide, nitrogenous base OH. So here we can easily define what is 5' and 3' end. See, this is nitrogenous base, this is nitrogenous base, this is nitrogenous base, this is nitrogenous base. Nitrogenous base can be adenine, thymine, guanine, cytosine. Out of 4, only one yeah, either it may be adenine, thymine, cytosine, or guanine. This is sugar component, this is sugar, 5 carbon, sugar, pentose sugar, we can say sugar, and this is phosphate group. See, in one strand we say that this is one strand, this is 5 dash, this is 3 dash. That means 5 dash means the phosphate group at the 5th carbon of the nucleotide which is present at the end, that is free. That means this is called as 5 dash end. This is called as 5 dash end. This 5 dash carbon bears phosphate group and this is free. And then we have 3 dash. What do you mean by this is 3 dash? This is 3 dash because 1, 2, 3 carbon group, hydroxyl group present at carbon. This is 3 dash end. This 3 dash end means hydroxyl group is present at the carbon third of the sugar, that is free. So we say that this is 5 dash to 3 dash. Another strand would be this. Another strand would be 5 dash here and 3 dash here. So we say that the two polynucleotides are antiparallel. This is at one end we have phosphate group free, at the other end we have hydroxyl group free. This hydroxyl group is present at the carbon third and phosphate group is always present at the carbon fifth of the sugar. So this is what is called as the two strands are antiparallel. This is what is called as, see, this DNA is a genetic material in all so DNA, it is a, this DNA, it is a genetic material in all organisms except riboviruses. Riboviruses are those viruses where RNA is a genetic material. What are called as retroviruses? For example, we have HIV virus, we have coronavirus, we have this tomato mosaic virus, we have tobacco mosaic virus. These are riboviruses where RNA is a genetic material. Where in case of prokaryotes, in case of all eukaryotes, we have DNA as a genetic material. This in case of eukaryotes, we have two types of. In case of eukaryotes, we have two types of DNA. One is called as nuclear DNA. We have nuclear DNA. Another is called as organelle DNA. This nuclear DNA, it is present in the nucleus, in the chromosomes. So this is present in the nucleus in chromosomes. And this organelle DNA, it is present in mitochondria. We call it mtDNA, mitochondrial DNA, or cpDNA, or called as chloroplast DNA. These mtDNA, that is called as mitochondrial DNA, cpDNA, that is called as chloroplast DNA. This mitochondria and chloroplast, these are called as semi-autonomous organelles. Semi-autonomous organelles. Why these are called as mitochondria and chloroplast is known as semi-autonomous organelle? Because they contain the genetic material. Then this DNA can be a single cell DNA, and then can be double stranded DNA. DNA is both single stranded DNA as well as double stranded DNA. This single stranded DNA is found in viruses. For example, we have 5 into 174 phage. 5 into 174 phage. It is the virus where we find single stranded DNA as a viral genome. As a viral genome, there is This DNA is single-stranded in case of 5 into 174 page. Then in all prokaryotes, eukaryotes, we have a double-stranded DNA. Then we have DNA, it can be cellular DNA. It can be linear DNA. DNA can be both linear as well as it can be circular DNA. Circular. What is linear? For example, I will show it like this. This is linear DNA. Linear DNA. And this is circular DNA. This is circular DNA. This is circular DNA. This is linear DNA. These are nitrogen bases. Nitrogen bases. Nitrogen bases. Nitrogen. This is. It would be like this. This is circular DNA. Circular DNA. These are nitrogen bases. Nitrogenous bases. Nitrogenous bases. Nitrogen bases. These are nitrogen. This is circular DNA. Circular DNA. This circular DNA, in case of circular DNA, the two ends of the DNA duplex are covalently linked to each other, so it becomes circular. The two ends of DNA duplex are covalently linked, resulting in the formation of the circular DNA. While as in case of linear DNA, the two ends of the DNA duplex are free. This is DNA duplex. This is DNA duplex. Duplex, it has two strands. That is why sometimes it's called a DNA duplex. In DNA duplex, where it is linear, it has free ends, and in case of where it is circular, the two ends of the DNA duplex are covalently linked so as to form the circular DNA. This circular DNA, it is found in where? It is found in prokaryotes. It is found in prokaryotes, and this it is found in nucleoid region. In nucleoid region as well as in the plasmids. This circular DNA, and this linear DNA, it is found in eukaryotic chromosomes. This linear DNA, whereas in mitochondria and chloroplast, we have also this circular DNA, whereas in nucleus of eukaryotes we have got what? Linear DNA. So the length, sometimes how you can define the length of a DNA molecule? Length of a DNA. How you can define the length of the DNA? Length of DNA. Length of DNA is the number of nucleotides which are present in it, or a pair of nucleotides known as base pairs present in it. So it is the number of nucleotides or number of base pairs present in it. Number of base pairs in DNA duplex. This is called as length of DNA. And this length of DNA, it is a characteristic of a species. For example, we have this. We have, for example, organism. We have type of DNA, and we have nucleotides or number of base pairs. For example, in case of 5 into 174 phase, DNA is single stranded DNA. Stranded DNA, and it is single stranded DNA. It is circular. Single stranded DNA, it is circular, and number of nucleotides, 5,386 nucleotides are present in it. 5,386 nucleotides are present in it. Same is the case with E. coli. E. coli, it has double stranded DNA, that is circular. It has 4.6 into 10 raised power 6 base pairs present in it, in its DNA. And then we have humans. How many humans have double stranded DNA? And this is linear. And how many base pairs are there? Approximately, it has been estimated, it is 6.4 into 10 raised, it is 6.4 billion base pairs. And in case of haploid human genome, this is 3.2 billion base pairs. And it has been estimated that there are, in case of E. coli genome, there are approximately 4,000 genes. And in case of this humans, we have protein coding genes approximately in between 40,000 to 80,000 protein coding genes in the DNA. So this was regarding the length of the DNA. And remember that this DNA is a dextrorotatory. DNA is dextrorotatory. That is, it rotates plane polarized light to the clockwise direction. That means it rotates plane polarized light towards right. So it is DNA is a dextrorotatory. Thanks.